to speak at this workshop. So far it's been a very impressive week for me. I mean, I've actually learned a lot of stuff, even though I work on the subject. So far it hasn't been too much on ChIP-Seq, so I've actually learned a lot of stuff myself. This is also my first time in Cambridge and at the Newton Isaac Institute, and I think this is really an amazing place. So the facilities are great, and I think a place is really serious about science and math when there's a blackboard in the bathroom. Okay, so as I said, this is going to be pretty different. So the good thing is that the previous talk I actually gave a lot of background on ChIP-Seq already, so I'm going to be able to skip a lot of stuff. Because this is only a 30-minute talk, I'm actually going to hopefully skip some details, but not too many details. So I will be hopeful that you guys will be able to interpolate in between my slides if some of the things are missing. If something's not clear, feel free to ask me afterwards. So here's a brief outline. I'll tell you a little bit about the ChIP protocol. So you've heard about using ChIP-Seq for nucleosome positioning via MNAs. Here we'll look mainly at transcription factors, so it's slightly different. I'll overview that. And then I will describe to you a pipeline that we've developed in my lab, basically trying to go from your aligned reads through the enriched regions where we believe there's a transcription factor that's binding, trying to find the de novo motif or the multiple motifs to form modules, and maybe trying to validate these looking at transcription factor databases and so forth. So at the end, I'll give you some references where to download the packages, some of the papers we have, and also some other stuff and material you can use to learn about the pipeline. So I should thank the next speaker, Peter, for these figures. So this is coming out of this review paper. So this is just to give you an idea. This is the DNA, and here you've got the nucleosome. So the DNA is packaged thanks to the nucleosome. And then it's accessible in between nucleosomes, so transcription factors can come and bind to the DNA. And using ChIP-Seq, you can actually try to interrogate where on the DNA these transcription factors or these proteins in general are binding. You can either look at transcription factors or at histones, which are part of the nucleosome. So you can either enrich or chip using chromatin immunoprecipitation by placing an antibody that will recognize that protein here, or maybe histone. But here we will actually mainly look at non-histone ChIP that in trying to enrich the fragments that have the transcription factor bound on them. And then what you do is that you'll fragment these, and then you're going to use sequencing to try to get the sequence and map that back to the genome to be able to tell here's the protein, the transcription factor that's binding to the DNA, here, here, and here, and so forth. So here's the main pipeline that we're using. So you start with your aligned reads, or you can align them. Actually, you can even do that in R. So we work mainly in R. So this is actually a pipeline that's written in R. I'll give you a little bit more details afterwards. So you go from here, your reads, you align to the reference genome. Then you're going to call your peaks. Then you're going to get a list of enriched regions. We're going to look for overrepresented DNA motifs where the protein actually recognizes and binds to the DNA. And then often what happens is that these tools will give you a very long list of motifs, and because there's a lot of background noise in these DNA sequences, what we want to do is be able to validate and maybe filter out the ones that we think are noise or background motifs or due to sequence repeats and so forth. And then in R, you can actually do a lot of stuff. It's very easy, for example, to look at the relationship to gene structure. You can visualize these. You can export and use your favorite tool afterwards. You can do a gene set analysis and so forth. So here, the backbones of our pipeline is going through peaks, RGADM for the motif analysis, and motif for the identification and validation. So I'll mainly talk about that. If you're interested about the other stuff, I'll give you some references. So here's the protocol when you're looking at a transcription factor. So it's very similar to the previous talk that when we've looked at trying to look at the nucleosome positioning, but here it's slightly different, mainly just the way we're going to fragment the DNA. Because we're looking at DNA-protein interaction, we don't really have to, in fact, you don't want to use an enzyme to cut the DNA. just because it's slightly better to use sonication in this case, that is just to vibrate your DNA fragments to break up the DNA into smaller pieces. So as a typical sequencing experiment, you start with, let's say, 1 million cells. What you want to know is where that protein is actually binding. So first you're going to cross-link the proteins to the DNA. Let's say here you're interested in the orange diamond. That's your favorite protein. You want to study it. The first step is that you're going to sonicate or fragment your DNA into typically from, let's say, 100 to kilobase pairs length fragments. You add the antibody that recognize that specific protein. You only want the fragments that have the protein on them. So you're going to enrich these fragments using chromatin immunoprecipitation and separate them. You end up with these fragments that only have the protein on them, the orange diamond. And then we don't actually care about the complex, so we reverse cross-link, we go and sequence, and then we're going to get our short reads. So for a typical ChIP-Seq experiment, you actually get single end reads just because it's cheaper, and in fact doing paired end reads don't get you something much better, but you can actually do it, and some people do paired end reads. So for nucleosome positioning, that might be worth it because you really want to know about a short events, that is where the nucleosome is actually binding, but for transcription factor, you don't really need to do that. As we've seen before, typically people will do a control, so they'll repeat the exact same experiment, but what they will do is that, for example, they will skip the immunoprecipitation step and they will just sequence the whole input DNA. Okay, so here's our genome. Here we've got 2 transcription factors or 2 binding sites for the same transcription factor, and let's imagine that after immunoprecipitation, this is what we end up for the fragments. So remember that what we're supposed to do is only enrich the fragments that are perfectly aligned with the transcription factor, because this is where the transcription factor actually binds on these fragments. But of course, there's some non-specificity, there are some background noise and so forth. So it's not uncommon to get a lot of reads that will fall off of where that do not perfectly align with the transcription factor. So this one is actually slightly less enriched than this one, meaning that probably in less cells the transcription factor was binding here. You either sequence the start or the end of the fragment, and that's sort of random the way that it's being done in the sequencing. So after you do the sequencing, what you're going to get are the row reads, that is, that read that come up from the beginning of the fragment, here, here, and so forth. So you're going to get the black and the red row reads, but of course you don't know if they come from the start or the end of the fragment, and you don't know where they come from on the genome. So the first step is to actually align these to the genome, and what you will get is a little cluster of black reads, a little cluster of red reads over here, same here, and then you've got some background reads. The other thing that happens, and this has been discussed several times already, is that some of the reads are not unique. So when you try to align them to the genome, you will get multiple places where they can map. So what people do in practice typically is that they will actually throw these reads away, and they say, well, we don't know where they align. They could align to 100 places. I don't know what to do with them, so I'm not going to use them. And in fact, often when you're given a data set, and if the reads have already been aligned, typically these reads have been removed from the data set already. Okay, so the first step is how can we get from these row reads and trying to quantify how much of the protein is binding at these places and these places, and also where the protein is actually binding. So what we wanted to do when we first started to work on this, and that was already over 2 years ago, is there wasn't too many methods available, so we wanted to use the bidirectional read. So we knew that there was some information that you wanted basically a red peak followed by a black peak, and we wanted to do something that's probabilistic, really model what's going on when the protein actually binds to the DNA in terms of the read positions. We wanted to use some prior information. So as you've seen in the experiment, what we do typically before ChIP-Seq is that we do a gel size selection. So we want to select the shorter fragments, and that's for mainly technical reasons, but also because having shorter fragments will allow you to separate closely adjacent binding sites. We wanted to derive measures of uncertainty for the binding site estimates, because we're statisticians, so we thought that was kind of nice to do that in addition to just having a point estimate. And the other big thing we wanted to do is that how can we deal with these reads that are missing? So, of course, we're not given the reads, but we know maybe where they will be missing, so maybe we can use this information to correct the density profiles for the missing reads. And so the methods we've developed for this, which is the first step of the pipeline, is called PICS for probabilistic inference of ChIP-Seq data. So the first step that we do in our pipeline is that first, as you can imagine, the protein will bind across the whole genome, but at just certain places. So the data is very sparse in a way. You're going to have lots of binding sites across the whole genome, but you're going to have lots of places where nothing's happening and the protein's not binding. So the first step that we do is that we try to cut the genome into places where we do get a lot of reads and maybe the protein is binding, and remove basically the places in the genome where there aren't too many reads that are aligned. And how do we do that? We just scan the genome with a sliding window, we count the number of reads, and then we join the sliding window to form basically a set of NGS joint regions, and then we're going to model these regions separately. So I'm going to show you one of these regions that we obtained after the sliding window. So this is really just a pre-processing step, if you want. So this is a picture coming from one of the data sets that we've analyzed. So as you can see here, these are the raw reads, so the aligned reads, and you've got your density of forward reads followed by the density of reverse reads. So if you sort of plot that with the density estimates, this is the dashed line, this is a kernel density estimate, you get a bump followed by another bump for the reverse. So the way we're going to model that, we say what are we interested in? First of all is the binding site location, which is mu, and then we know something about the fragment size, so we're going to model the average fragment size for this as delta. And then what we thought maybe would be a good model is to have something that's slightly symmetric for the forward read positions and the reverse read positions with different variances. And because there are some outliers here and there, we use a T4 distribution, which is slightly more robust than the Gaussian distribution. So the forward distribution is centered at mu minus delta over 2 with variance or variance parameter sigma f squared. And then we've got another T distribution that's centered at mu plus delta over 2 and variance parameter sigma r squared. The good thing about this model is that we remodel what we care about, the binding site position, the location where it binds in the genome, and the average fragment size distribution, something that we know about, and therefore we can sort of build a Bayesian model, put some prior information into that. Yeah? Oh, so that's a good question. So I'll get back to that. It's basically here you cannot align any reads because it's highly repetitive. So I'll get back to it when I'll tell you how we deal with this. Okay, of course, when we do the pre-processing, it's very likely that you're going to obtain regions that have multiple binding sites in them because maybe they are very close in the genome. So what we do is that we can actually generalize that using a mixture models and trying to separate the binding sites that are close to one another. So here, this is how we use the prior distribution. So often when you do these kinds of experiment, you're going to do a gel site selection. So you can actually get an idea of the distribution of the fragments and sort of get a pretty good range for your fragments. So what we do, and this is really for computational reasons, we use normal inverse gamma prior, and we're going to set these hyperparameters so that the fragment size distribution really fits what what's been selected for the experiment. So I'm not going to tell you too much about the parameter estimation, but it's actually pretty straightforward. You can derive an ECM algorithm with closed forms, E steps and M step, and that's why we use these conjugate priors, just because it makes the computation a lot easier. Remember that it's very common to have, let's say, 50 million reads in these data sets. So we wanted something that we could code efficiently and that could actually run in these data efficiently. Okay, so let's get to something that might be slightly more interesting is how do we handle the missing reads? So we know that there are reads that are missing because they couldn't be uniquely aligned to the genome. But we don't know which reads are missing, right? However, we know where they might be missing because we know the genome, we know the reference genome, so we can sort of scan the whole reference genome and try to guess where the reads are missing because it's highly repetitive. So this is what we do basically. We're going to use exhaustive enumeration, enumerate the whole genome, try to look at where it's highly repetitive, and generate what we call a mappability profile that will tell you here you can uniquely align a read, so we put it 1, and 0 where it's impossible to uniquely align a read because it's repetitive across the genome. So at the end, we generate these 0, 1 mappability profiles. In fact, all we care about here is where we cannot align the read, so only the 0 profiles. And if you want, it's very easy to convert that into a bed file, and then we use that for the computation, where we know that we cannot align any reads. So here's an example of the profile. So this is the density. You can see that you've got your forward reads followed by the reverse reads. This is the profile. So this is the gray rectangles. And here you see you've actually got 3 disjoint intervals where you virtually get no reads in there. Of course, it's possible that you get a few reads once in a while because here the aligners allow for mismatches. So sometimes, we might not get the exact representation of the profile. But it's a good approximation for us that we can use. So how do we deal with that? So we don't know what reads are missing, but we know basically where they should be missing. So given the density, so once you estimate the density, if you know the density, you can sort of estimate how many reads you should have observed in your intervals where there's no reads. And basically, given that number, you can sort of estimate the contribution of the reads for the density profile. And in a way, it's very easy to incorporate that into an EM algorithm. The E step just estimates, how many reads are missing, what their contributions are. You impute that, and then you carry on with the M step. And what's very good is that you can actually get closed form estimates again for the E and M step. So it was pretty easy to incorporate. And I say pretty easy because it's easy to get something that's closed form. There's actually a lot of computation because you deal with truncated distributions. So here's a concrete example of what this does. So this is a stat1 data set. There's a motif here, so we know that this is where the protein should actually bind on the DNA. If we don't account for these profiles, so if we just say everything's fine, nothing's missing, and we estimate based on our model, this is the estimate that we get. So this is the cross. And the brackets give you the plus or minus 2 standard errors. So I'm not going to say this is really confidence interval, but that gives you an idea of the variability. And here we can say that we're actually off the estimate. And when you correct for the missing read, you can see that it shifts on the left, and you'll basically sit right on the DNA motif. OK, so this tells you how we estimate the binding site locations. Of course, when you're going to scan the whole genome, you're potentially going to get lots of false positives as well, right? Because we're going to scan the whole genome. If you've got a few reads, or maybe if the profile is not very good, you're going to get some false positives. So the way we're going to deal with that is that we're going to score each region, and then you're going to get a ranked list, and maybe based on some kind of threshold, let's say an FDR or something, you can threshold your list and say this is what I believe where the protein is actually binding. And the way you can estimate the FDR, you can just, in the case you have a control sample, you can swipe the IP and control samples, you can rerun that, and you can estimate an FDR based on that. Okay, so this is an example of an FDR plot that's generated with our ChIP-Seq package. This is the FDR, this is the score, and that gives you also the number of regions that you get. So here, the gray line is the 10% FDR, and that will tell you that maybe 15,000 regions are called positive in that case. So this is a couple of examples that I wanted to show you. This is just to show that actually the mixture model is pretty good at separating closely adjacent binding sites. And in particular, it's good at that because we use the prior information on the fragment length distribution. So we know roughly what the separation between the forward and the reverse peaks should be. And therefore, we can use that to sort of put more information into separating 2 peaks that are close to one another. So this is an example. This is the peak score profile. This is just using the combined forward and reverse density profile. It's just easier to visualize one track than 2. This is again using the genome brother, UCSC genome brother. This is a FOXN1 data set. What I show you here, so you can see the profile. You see the aligned reads. So blue is forward, red is reverse. So you can see there's a stack over here, another stack over here. There's actually 2 verified FOSA1 binding sites here. And you can see that peaks will separate them into 2 enriched regions. This one has lower score than this one, so that's why it's in gray. And here, this is actually another peak color, which is called max. It's pretty popular But the advantage of PICs is that typically you will get short enriched regions and you will separate the closely adjacent binding sites. So this is another example where you've got two CTCF binding sites that we separate into two, Max doesn't. Here another example, actually we got three in this case that are separated into three separate enriched regions. And here another more extreme example where it's quite messy. There are a lot of reads that are stacked up. But in fact, what we do is that PICs separated into two different enriched regions, and you've got two binding sites in these two. So they are somehow validated. Another thing I should mention is that the pipeline that we work with actually deals pretty well with repeats. So even though here there are a lot of repeats as notified by repeat masker, the pipeline can work well with this. Even the de novo motif, we don't filter anything, we don't mask anything, we just run everything, and then we post-process and validate. And I think this is actually pretty powerful. Okay, so this is another tool that we've developed, and I say this is really a tool because the methods was already existing. What we done that we've collaborated with someone trying to improve it, especially for ChIP-Seq, make the software a lot better, run in parallel and this sort of thing, so that it's very efficient to work on large ChIP-Seq data sets. And my philosophy is that if you think there's a software that works pretty well or a method that works pretty well, well, you know, don't reinvent the wheel and just try to use it. So here, this is what we've done. You know that there exists pretty many softwares for the analysis of de novo motif, but in fact, many of these were implemented, let's say, maybe 10 or 15 years ago when there was only a handful set of sequences. So maybe at the most you got 100 sequences, and it was very easy to do the de novo. And one popular tool is MIMI, for example, that a lot of people have used, but it's extremely slow. If you want to use it on a full ChIP-Seq data set, even on a cluster with 24 nodes, that might actually take you several weeks. So it's very, very slow to run on a very large data set. There are other softwares that are sort of faster, but these are based on enumeration of k-mers, and sometimes they might not be as accurate as probabilistic methods. So what we've done is that we've worked on GATM, and I will tell you a little bit about it, which was published in 2009 in the Journal of Computational Biology by Leping Li. And we've tried it, and we thought it was quite good. It was working very well. It was fast. It was finding a lot of motifs. Some of these, a lot of these we could validate. So we say, well, let's try to get an R package that will build in the pipeline and make it even more efficient. So RGATM is really fast, it's probabilistic, and can detect multiple motifs. It will detect motifs, mask and repeat, mask and repeat. So why is it called GATM? Because it uses a genetic algorithm and followed by an EM algorithm. So it enumerates a bunch of k-mers, form space diodes, and then use a genetic algorithm to only retain a few of these. And at each iteration, also uses an EM to improve the PWM. And at the end, it will match the motifs that it found and repeats and so forth. What we've done here is that really optimize and provided more options. So the next step is, once you get your list of motifs, this is an example. So you've got multiple motifs here. I only show three of them, but on a very typical ChIP-Seq data set, you will get maybe from 30 to 100 motifs. And what you want to do is, first of all, identify these motifs, see if this is something that was already known. Maybe it's in a database somewhere, and maybe this is something that you really know, or you expected, or you know that actually collaborate with the primary motifs, the one that's the protein is known to bound to. So what you will do is take your list and maybe use a database to try to identify the motifs. Okay, and if you've tried to do that before, either you will go through a web interface where you need to input one PWM, get the output, validate. So if you've got 30 or 70 motifs, it can be pretty slow. There are tools to do that, such as Stamp, if you've used them before. But again, this will give you like a PDF printout with maybe 70 pages that you need to go through. So that was pretty long to do that, and it wasn't flexible enough for us to work on very large data sets. So what we've decided to do is to work on a tool very similar to Stamp, but with more features, really rich graphics outputs, and a lot of filtering things that we could do. So this motif that we derive can actually be used to compare the list of identified motifs to a known database of transcription factors such as Jasper or TransFac, or maybe your own database. You can actually input it in the software and use that if you want. By default, Motif actually contains the 2010 Jasper database, which is probably one of the best databases for transcription factors anyway. It also contains pre-computed scores based on this database, so we can compute e-values and do all the alignments for you very efficiently. We've got grid-like graphics capabilities. You can do motif matches. You can look at distance and pairwise distance plots. And what we think is very important, you can do the filtering to select only the motifs that you think are relevant for your experiment. So here's an example of an output from Motif based on the Jasper database. And what we've done here straight off and say, okay, let's only look at the one that have a good match to the database. So we use an e-value cutoff of 10 to the minus 4, and already this sort of filters out only 8 motifs that might be more interesting. And in fact, if you look at this, you will see that this is a stat1 experiment. The first one that you find is the match to the stat1 motif. Another one that you find is the AP1 motif, which is actually known to interact with stat1 in this case. And another one that we found with an extremely good e-value is the CTCF motif. So something that we couldn't really find in the literature that was known about the interaction of these two proteins of stat1 and CTCF, but it was clear that there was an enrichment for the CTCF motif here. So the next step is, how can we validate these? I mean, sure, you find a good match to the database, but it could still be that this motif is actually just a background, and it's just because maybe you're looking at a lot of promoter regions, and promoter regions are very rich in all sorts of motifs. So how can we further validate these? So what we decided to do is that in addition to just look at the motifs themselves, maybe we can look at the distribution of the motifs within the enriched regions. Okay, so here's an example that what we can do is that this is the exact same plot, but instead of looking at the match in the database, we're going to look at the distribution of the motif occurrences within the enriched regions. The idea is because the enriched regions are centered on the prediction from peaks, so the prediction of the binding site, not looking at the sequences, just looking at the reads themselves, is that if the motif is valid and has a preferential attachment to the primary binding site, they should be pretty well centered around that region. Okay, so this is the case for the stat1, the CTCF, and the AP1. You can really see you've got a peak distribution around the center. If you look at the other motifs, it's rather flat and tells you that there's no preferential positioning of that motif across the enriched regions. So we tend to believe that these are probably background motifs and they're not interesting. So then once you've selected these motifs, what you can do is that you can actually do pairwise distance plot. And again, these are all built-in functions. You just type one line, it does it for you. So you can look at the stat1, the distance between the stat1 and the AP1 motif occurrences, and it does that in sort of a 2x2 table. Same stat1 CTCF, AP1 CTCF, and you can see here it's sort of really centered around 0, meaning that the motif occurrences are pretty close to one another. So there's potentially a collaboration between the two proteins. Same for stat1 CTCF, and you really see that bimodal distribution here. And also, these kinds of plot will not only give you the distributions, but also the number of motifs that are found and the overlap with the percentages. So this really tells you, how much motifs you find that are together, the distance between them, and so forth. Okay, so I'm pretty much done. So I'm going to tell you a little bit more about the R packages, give you some references. So the first thing is that what we've tried to do is to really write these tools that are very efficient. So all the core functionalities, all the main computations are done in C. It's wrapped in R. We've tried to use modern technologies such as OpenNP or Grand Central Dispatch on Snow Leopard on Mac OS X, or Snowfall and Multicore. So it depends on what tool we're using and what the easiest thing to use in that case. It's also class and method oriented. So as I told you, it's very easy to do these plots. You just type plot and the name of the object, and depending on the type of the object, you will know whether to do a motif match plot or a distance plot or a pairwise distance plot and so forth. It's very flexible, meaning we've got peaks, argat-m motif, but if you've got your own peak finder, you can just use the other two. If you've got your own motif finder, you can just use motif and so forth. And the great strength about these packages and Bioconductor and R in general is that you can easily communicate with other packages. So in particular, you can do annotation of these peaks with ChIP-Seq. You can do further sequence analysis with biostrings. You can use R-Track layer to communicate with the UCC genome browser. So all the images I've showed you in these slides, I actually use R-Track layer. I never use the web browser. It's just communicate with it directly from R. So here's a paper that's in Press in Biometrics that sort of describes the PICS methodology. We've got an article describing the pipeline that's been submitted. I'll be happy to send you a copy if you send me an email. We've got the R pipeline, so PICS, RGADM, motifs available from Bioconductor. I didn't talk about ChIP-Seq, but you can actually use RMAT or a package to do ChIP-Seq if you want, and then use RGADM and motif. I gave a course not long ago at Cold Spring Harbor describing these packages. So there's actually, I built a package with a bunch of data and a demo in it. You can go and download that. There's the SVN if you always want it up to date, or you can just download the tarball. There's a lot of good stuff in it, and you can really follow the tutorial. So it was a sort of a one-day course on it, so there's a lot of material in it. For the people who are staying here, I'll be happy to give a demo if you want, and going through the package and stuff, or some of the other functionalities. People I should thank, so really the people who did the work. So Xu Guizheng did the work on PICS with help from Kite and Ning, looking at other softwares. Arnaud Droit and Eloi Mercier actually developed the RGADM and motif packages. We've got collaborators at the Genome Sciences Center and the Clinical Research Institute of Montreal, and some people that have contributed to some of the data and the funding. Thank you.
Thank you, Raph. Are there any questions? Hi, Corny.
So I guess this, you're putting down these sequences on the basis of an antibody having a bound to the protein.
Yeah.
Now it's possible, I guess, that antibody could bind to a number of different proteins. And so you're going to get different motifs apart from each of the locations that you find these things. Is there, I mean, is this kind of relevant, or is there some sort of way in which you would want to sort of synthesize these different results from these different?
Yes, so it is relevant. I mean, the first thing is that antibody specificity is one of the biggest problems in ChIP-Seq, because, you know, there's a lot of proteins that people still can't interrogate because we don't have the antibody for it. And there's a lot of cross-binding, so it binds to another protein. So it could be that some of the motifs that you get actually due to these things. We believe not, because when we look at the overlaps, we can see, well, there is the primary motif that we expect, but there's another one that's next to it. So we do find the primary motif, the one which should bind, but we find another proteins that's next to it. Then there's also some, you know, proteins that bind directly to the DNA. So there's a protein that binds to the DNA, the protein binds to that other DNA, and that's actually the primary protein that you're tagging the antibody. So you're not going to find the motif there, but you're going to find the motif of the other protein. And we do find some of these things here, and in fact, it's known that the interaction of type 1 and AP1, sometimes that happens. There's AP1, there's type 1 sitting on top of it.
Questions? Yes.
It's great to see a pipeline that goes all the way from taking the data off the instrument all the way through some downstream analysis. Those kind of tools together are extremely useful. I had a question about the where you're estimating the reads that that can't be mapped to the repeat regions, and you have an interesting way of estimating it. Obviously, some of the aligners can tell you the reads that could have mapped. So instead of throwing them away, you know, hey, you know, this read could have mapped to these 10 places, and it's really just a matter of narrowing it down. Have you looked at all about taking the information from the aligner about?
Well, we haven't looked. I mean, the first thing is that when we started to work on this, that these aligners didn't exist. And then, even if it tells you, well, it could map here or could map here, if it gives you, you know, maybe 1,000 places for the read where it could map, well, it doesn't give you much information, right? So in a way, what we do is that we look at the profile and say, well, let's just look at the neighboring profile, and that's going to give us some information about, you know, how much or how many reads we should have seen in this interval. So you could potentially bring in more information by saying, well, you've got some of these probabilistic reads that says, well, there's 1 out of 10 match here and so forth. But we haven't really looked at it just because we think it works just well like that. But potentially, yes.
Thank you, Kendra.